Okay, guys, pardon. Okay, so I will give this big presentation. I separated it to two parts just because it is more than 1 hour in total. And I give more information in this presentation, and I give references, references to the source of information. So it is big presentation. So, but it is more details, more explanation. So probably you will get more help from this. So the question is, should you choose to live forever? It's possible. Okay. So again, you can download the presentation as PDF file from the link in the description. Then you can go to the links, click on the link, and then go and understand maybe better. So let's continue. In my recent video in presentation, I mentioned that snake mole rats are not aged to live about 40 years compared to wild rats and mice that typically live less than 2 years, mice actually 1 year. Most researchers explain extreme longevity as evolutionary adaptation to live in underground and high antioxidant activity in snake mole rats. Cancer development in mice and rats is a major cause of death that significantly reduces their longevity, and in contrast to mice and rats, snake mole rats and long-living blind mole rats are remarkably cancer resistant. Along with most other mammals, snake mole rats and blind mole rats are able to synthesize vitamin C within their bodies, and vitamin C plays a crucial role in various body functions, primarily as antioxidant, and cofactor is an essential process in our body. So you see as this slide that snake mole rats live up to 40 years. It's actually maybe longer because they're not aging at all. Blind mole rats, we have less information, but it is known that they live about 30 years. So another mammals that live long, it is bats, and some microbats, they live up to 40 years compared with the wild mice of the similar size that lives only 1 year in total. Vitamin C is vital for growth, development, and repair of all body functions, tissues, including the formation of collagen and elastin, absorption of iron from foods, immune system function, wound healing, and maintaining cartilage, bones, and teeth. Industrially produced ascorbic acid has 2 mirror image isomers in equal parts, commonly denoted as L and D. L, it is true vitamin C, and D has no significant biological role. Linus Pauling mentions that animals typically produce 10-12 grams of vitamin C daily, I mean, to body of the humans, compared to body of humans. And suggested intakes of large doses of ascorbic acid up to 18 grams daily, because you need twice more of the ascorbic acid to get a necessary amount of vitamin C. So instead of officially recommended 75 to 90 milligrams for adults, which is not sufficient at all. In his later years, he followed a low protein, low sodium diet as recommended by his physician, and also he was using a vegetarian diet. And he lived only for 83 years. This again demonstration how it is dangerous to follow medical professionals. It is absolutely, I don't know why he did it. It was so easy. In 30s already, it was quite well known that the use of carbohydrates as a source of energy leads to development of cancer, and this is what happened with him. Okay, so in normal cells, vitamin C works as antioxidant mostly. In cancer cells, it works both as blocking glucose transport to the cancer cells, and also it is working as pro-oxidant, and as megadoses of vitamin C may lead to cancer death. Megadoses of vitamin C may reduce glycolysis in hyperactive cells. This is transport because of reduced transport of glucose. And they enhance apoptosis in senescent and cancer cells. Okay, you can read in more details in the publication. Pharmacopeitical studies show that intravenous administration of sodium ascorbate may result in much higher, up to 70-fold concentration in the blood than after oral intake, and that such concentration is useful to treat cancer. Pardon. A dual combination of doxycycline with vitamin C was investigated as new synergistic metabolic strategy for starving and eradicating cancer stem cells. And again, it works quite well. A triple combination of doxycycline, azithromycin, and vitamin C. So in this case, doxycycline and azithromycin much smaller doses than doxycycline alone with vitamin C, and it works much better. But it was investigated only in laboratory cell cultures as senolytics, which selectively kill and remove approximately 90% of senescent fibroblasts. Personally, I tried this combination; it works. Okay, so again, there should be some clinical studies, but nobody pays for this because this doxycycline, azithromycin are easily available, vitamin C also, so nobody will pay for this antibiotics and vitamin C, so there is no profit from this research if it will be clinical trials. Clinical trials are very expensive. Okay, so you can watch my videos for more details. Okay, this is again one more presentation showing how this combination of vitamin C with antibiotics work. So okay, you can again go and check it. It is very interesting. Again, all this it is to cure cancer. It is relatively easy. There are other methods which are not only following ketogenic diet or the use of these mega doses of vitamin C with antibiotics. There are other methods to suppress cancer development. But curing cancer, it is not curing cancer. Medical professionals don't cure cancer. They just stop propagation of the tumor. But then, anyways, the people don't live too long because after chemotherapy, chemotherapy and other interventions, people don't live long. Maybe 5 years, the most, the longest after these treatments. Okay, I discussed the radical life extension for humans in my old videos. You can check them. And then social insects such as honeybees, ants, and termites demonstrate staggering lifespan differences between queens and working members of the colonies despite having the same genetics. Again, so genetics cannot change you. If you try to change your genetics, you will only shorten your life. Trust me, again, it is, you are not, so you are not supposed, your genes are already optimal. During their life, queens don't consume plant-based foods but substances like royal jelly, larva hemolymph, and termite jelly, royal jelly. So it's saliva produced by worker termites. So it's no plants. It is how to live longer. Don't consume any plants. Many plants have toxins known as allelochemicals produced as a defense mechanism against herbivores and insects. Some fruits and veggies are also high in oxalates that increase the risk of kidney stone formation and other problems. Consumption of some plant-based food may be critical. So avoiding consumption of some plant-based foods may be critical for longer life. I don't say all plant-based food are bad, but mostly yes. This is long-living insects, termites, and honeybees, and the ants, and wasps. Wasps are not living too long because the colonies are small, and the wasps, they are using not plant-based food. They consume some substance which larva produces. Larva produces kind of milk. And both the queen and the working members of the colony, they consume this milk. They don't eat insects. They don't eat plant-based food. So, and this, but I don't know about how long the queens in wasp colony lives compared to working members. Naked mole rats and blind mole rats rely on the consumption of the interior flesh of the large tubers that is rich in raw starch and low in toxins, which is the point. Again, the interior flesh of large tubers is low in toxins. All toxins are in epidermis. So there is other explanation why they not consume epidermis, but again, I believe that they understand that epidermis is high in toxins. And again, if you eat potato, for example, it used to be so toxic, but now it is genetically a little bit modified. It is selected so that it is not as toxic as it used to be. And but anyway, it is again, tubers are mostly toxic. Even cassava tubers and potato tubers, they are toxic. Inside, yes, inside they are less in toxins. Hepato raw starch cannot be digested and called resistant starch. So, and bacterial fermentation of resistant starch in cecum and large intestine produces short-chain fatty acids and other metabolites like vitamin C, which potentially influence longevity in naked mole rats. Okay, so you can read it. So again, this is explanation why the naked mole rats live longer. In aging humans, researchers observe higher reliance on anaerobic glycolysis compared to fat oxidation for energy production, compromised digestion of protein, degradation of bones and muscles, and body fat accumulation. In contrast to aging humans, in older naked mole rats, researchers observe higher reliance on lipid oxidation for energy production, and high lipid metabolism is involved in social status transfer from worker to breeder. So young naked mole rats, they consume this raw starch, they consume interior of flesh, but the older individuals, they consume actually more bacterial product of bacterial fermentation of these tubers. Okay, this is how it can work in our body. Starchy food. It is, but mind that it is, if it is cooked, then resistant starch is very small portion. So mostly it has to be different. This has to be, big and this small. But okay, if you take, let's say, raw starch, then some amount of the raw starch can be digested and converted to glucose. The rest is resistant starch, and resistant starch is very beneficial for butyrate production and other short-chain fatty acid production. So what is happening during the aging in humans, that the people, because of development of chronic hypoxia, the cells rely on anaerobic oxidation, so without oxygen, not oxidation, but anaerobic glycolysis, fermentation of glucose to lactic acid. And then it is very low efficient process. This is why all the people have low energy. And then mitochondria get damaged because of acidic environment, also because of higher production of, because of oxidative stress and higher production of reactive oxygen species. So this is what is happening. So you have to, in old age, you have to rely on lipid oxidation. Of course, it's not easy. Adaptation in old age to ketogenic diet takes long, maybe 4 months, maybe longer. It needs some, you can find out in other people also talking about how to improve this and accelerate this transition to ketogenic diet. In particular, you have to consume more electrolytes. The naked mole rats possess a unique gut microbiome composition that resembles the healthy microbiome of centenarians and hunter-gatherers. So it means more beneficial bacteria than pathogens. Similar to rabbits, naked mole rats practice coprophagy, which is the act of eating their fecal pellets. Fecal pellets, it is these tubers, result of the bacterial fermentation of these tubers. In Naikmo-Reds colonies, social roles are divided based on age. Young individuals primarily are focused on digging burrows and food delivery, while larger, older members specialize in defense. Transport of food through narrow burrows is complicated, therefore all the new Naikmo-Reds rely more on consuming fecal pellets of younger Naikmo-Reds that are enriched with short-chain fatty acids, amino acids, and vitamins group B. Amino acids like alanine, aspartic acid, glutamic acid, isoleucine, leucine, serine, and threonine. 309 are more abundant in fecal pellets of Naikmo-Reds compared to the feces of other rodents like mice and especially mice, rats, and Western humans. Okay, so at least it gives you some idea about what to eat. And also epithelium layer in Naikmo-Reds are much thicker, and the cells, goblet cells, which produce mucus that protects the layer of epidermis, they are much more active, much larger and much more active. So it is again Naikmo-Reds. You see the secret of their longevity. They live long because they have very healthy epidermic layer of epidermis in the gut. Okay, this is the secret. So there are abundant short-chain fatty acid producing bacteria in the gut microbiota of Naikmo-Reds. Short-chain fatty acids such as acetic acid, propionic acid, and butyric acid, and their esters have remarkable health-promoting and anti-cancer properties. Butyrate improves intestinal epithelium cells' junctional integrity, which prevents bacterial and toxin penetration in the remote organ of the host. That has been suggested as a potential factor in longevity of Naikmo-Reds. Again, scientists understand that longevity of Naikmo-Reds is related to this production of short-chain fatty acids and related to health of the layer of epithelium layer in the gut. Similar to younger Naikmo-Reds, blind mole-reds rely on consuming tubers which are rich in raw starch, but a part of raw starch is digested and converted to glucose in small intestine. The rest is fermented by bacteria to give short-chain fatty acids and other metabolites. And because of glucose produced in the small intestine, black mole-reds generally have more body fat than older Naikmo-Reds. This is again okay. You may see, you may check the photo of blind mole-reds. They are really thick. And my personal experience also shows that when I consume too much of this raw starch, like more than 200 grams of raw starch a day, I started to accumulate subcutaneous fat. So it's again, and my weight went up. Okay, so I reduced it to 100 grams. Then okay, that you have to understand. So carbohydrates such as resistant starch, they get converted to butyrate. And the only dietary fiber which is good for your gut is resistant starch, nothing else. Okay, this is, you have to understand, nothing else works. Okay, I will talk about this later a little bit. Butyrate, which is generated by fermentation of resistant starch in the cecum and large intestine, is particularly important in maintaining health. Again, secret of the longevity. In contrast to Naikmo-Reds, both mice and humans show age-related shifts in the gut microbiota, a decrease in beneficial bacteria responsible for production of butyrate, and increase in pathogens. Okay, everything is clear. There is increased evidence that gut microbiome plays a crucial role in longevity across species and the development of age-related health program. Again, people, scientists understand. There are some claims that to year 2030, the secret of internal life will be open. I do it now. Okay, it is now 25, so you know already. Pardon, I'm in village and it is sometimes very noisy. Okay, so you see the secret. Age-related changes in the microbiota have been strongly linked to increased intestinal permeability, or so-called leaky gut syndrome, pathological activation of immune system, because these pathogens and also bacteria and all the microbes get to the bloodstream, and then immune system is activated, and overload of the immune system leads to immunosenescence and other age-associated chronic inflammation in the host. Okay, this is all related to bad diet. You see, how to make it, you live long, you have to consume proper food. So eventually this leads to aging, accelerated aging and death. This is what's happening. Okay. So Western diet, yeah, this is why you live so short. Okay, this, if you rely on medical, on recommendation of official medical professionals and dietitians, you are not supposed to live longer than 100 years, maybe 90 years the most. So, but yet again, it is okay. It is okay. To me, it is okay. I am not following recommendations of the medical professional. I am not sick. Last 25 years, sometimes I make mistake. October last year, I made big mistake. I made a vaccine, not against COVID, but anyway, I was thinking traditional vaccines are relatively harmful. No, I got big complications. So I'm so angry on me, on myself, that my stupidity to allow to make this vaccine. Okay, let's go. Maybe I talk about this because it is again situation. It relates to what's happening in Thailand, and not only in Thailand. In China, you know, the outbreak of the COVID, and in Thailand already people are thinking about next lockdown, that maybe there will be a next lockdown. And why it is happening? Okay, I will not talk about this because it is a little bit dangerous. My videos were deleted from YouTube when I would try to explain it. NECMO-RAD's microbiota showed an abundance of bacteria able to use sulfate, sulfite, and other sulfur-containing molecules, which are important to produce these antioxidants. Although the diet of the NECMO-RADs have approximately 10 times lower levels of total proteins than that of mice, but they can get protein from fecal pellets that have approximately 20-24 grams of protein per 100 grams of microorganism. So it's, again, you see the way how to get protein into your diet, and this is, I should work on it probably. So we have to maybe think about production of protein, how to make a biological, chemical reactor, biochemical reactor to produce all this protein and amino acids and etc. from carbohydrates. I buy amino acids which are produced industrially from carbohydrates, but without protein. They are already purified. But probably it is a good idea to make such a reactor at home and produce yourself from carbohydrates like sugar or some starch or whatever. The levels of glutamate, cholesterol, and bile acids are significantly higher in NECMO-RADs compared to mice. Again, you see, glutamate is, I was talking about this, glutamate is very beneficial. Cholesterol, we need a lot of cholesterol because cholesterol transports the fat from the gut to the organs, to the liver first, and then liver makes low density lipoprotein to transport these fatty acids further, etc. So you need, and they rely on fat oxidation, not on glucose. And bile acids are absolutely important for detoxing of your body. So okay, this was again compared to mice, it was much higher concentrations, so you can learn what you have to do. Glutamate is important to transport ammonia from guts, muscles, and lungs to the liver to be converted to urea and excreted by kidneys You can watch my video for details. It is very easy. Glutamate can work as a fuel for layer of epithelium in the gut. But when it is consumed, then it is producing ammonia inside the cell. But also ammonia can be produced in the lumen of the gut. So glutamate is important because glutamate reacts with ammonia, of course with some enzymes, participation of enzymes. Then glutamate get converted to glutamine, and glutamine already go to blood and transport ammonia from the cells in the lungs and muscles to the liver, and in the liver it is ammonia converted to urea and excreted by kidneys. Okay, you can watch more my video and for more details. Cholesterol is necessary to transport lipids in the blood plasma and produce hormones and bile acids again. You see, absolutely important. People, medical professionals call so-called bad cholesterol, and they say cholesterol is harmful for you. It is not so. They call low-density lipoproteins, they call bad cholesterol, but this never gets bad unless you consume a lot of polyunsaturated fatty acids. Polyunsaturated fatty acids in low-density lipoprotein is making low-density lipoprotein bad. But they call it bad cholesterol and blame all cholesterol. Cholesterol is a little part which is necessary for stability of membrane of the lipoproteins and the stability of membrane in cells, and for production of lipids, for production of bile acids, for production of hormones, pardon. Okay, consumption of animal fat rich in cholesterol can be beneficial for health. See my video and presentation, so you can go and check it. Yes, so I follow ketogenic diet, but actually it is carnivore ketogenic diet with a lot of animal fat, mostly butter. Okay, I will talk about it later. So you see how it is glutamate gets converted to glutamine, and glutamine goes to liver, and then in liver ammonia gets converted to urea, and urea is removed by kidney, and but then it is converted again to glutamate. This is wrong. People blame glutamate. They never say glutamate is good. Glutamate is very essential part of our diet, and if you want to live long. But they blame it. They blame like cholesterol. They blame saturated fat and animal fat as bad fat and source of cholesterol, and they blame glutamate, in particular monosodium glutamate, as dangerous. It is so far from reality, so far. I don't know how people trust medical professionals. I don't understand at all. If you go to science, you see absolutely opposite works. With aging, gut transit time tends to slow down, particularly in the colon, perhaps due to a more refined diet and reduced physical activity in older age. The longer colonic transit time have been associated with reduced short-chain fatty acid concentration in feces and increased productions of phenols, sulfates, ammonia, and trimethylamine, which are very toxic, and due to degradation of undigested protein in the colon. Okay, you mind that in older age you cannot digest protein properly, so the only way what to do, remove this undigested protein, so make your short gut transit time very short, that even if not all protein consumed, okay, get it out, just get it out, like microbes do. And what else? You have to keep pH, acidity in your gut, such that it should not be alkaline, it should be acidic, slightly acidic. So a diet rich in butyrate generating resistant starch shortens colonic transit time in rats. Yeah, so because acidity is because of acidity. Acidity ignites peristalsis, ignites and accelerates the transition of food through the gut. Increasing the content of resistant starch in daily diet would be beneficial for maintenance of gut health and reduce risk of colorectal cancer development. Okay, it's again already known, but how much? It is recommended maybe 50 grams of resistant starch, not just starch, raw starch, resistant starch, 50 grams. It is quite a lot, I would say. And if I consume 100 grams of raw starch, it is only 70, maybe 80% of this resistant starch, so 70, 80 grams. It is more than recommended, but again, I see improvement. If you take too much, then as I said, then too much of starch converted to glucose, it is already not good because you have to stay and I will talk about this a little bit later. Dietary interventions may help to reinforce intestinal integrity, target age-related dysbiosis, reduce uncontrolled activation of immune cells, prevent immunosenescence, and suppress systemic low-grade inflammation. Again, guys, in scientific publications, everything is clear. You just read, you have to understand it, and then all the picture is clear. So this is how pH is changing in the gut, and what is the transit time. In the colon, it can be 70 hours. But they call it neutral, 6, means neutral, 6 to 7 is neutral, but sometimes it is alkaline. When it is too long, it is getting alkaline. But it has to be slightly acidic, slightly, not much, slightly acidic, maybe 5.5 or maybe 6, like this. This would be, this will shorten the colonic transit time. And diversity is getting larger, and with colonic transit time shorter. The shorter, the more, okay, pardon, I confused you. Here, short colonic transit, this is slow. With short colonic transit, microbiota diversity is small. And somehow medical professionals claim that it should be diversity, as you need diversity of microbiome. You don't need diversity, you need beneficial, friendly bacteria. And this number of these bacteria is small, tiny. So you don't need diversity with all these pathogens, etc. No, you need only friendly bacteria. Friendly bacteria, they produce short-chain fatty acids. Pardon me. They produce short-chain fatty acids, and they accelerate colonic transit time. Okay. In aging humans, both the mass and number of mitochondria in muscle cells tend to decrease compared to young individuals, leading to a decline of muscle function and strength. Studies have shown a 50% reduction on oxidative capacity per muscle volume and 30% reduction per mitochondrial volume in elderly adults. Again, why? Because you cannot digest enough protein. If you have no amino acids, then your muscles are consumed and bones are destroyed, because bones also a source of amino acids. The function of mitochondria, including the abilities to produce energy by aerobic oxidation, the number of mitochondrial cells, and the control of reactive oxygen species, are compromised in skeletal muscles of aging humans. Again, so because of chronic hypoxia, because of chronic inflammation, because of high oxidative stress. In contrast to humans, the mass and number of mitochondria in muscle cells on neck mole rats is increasing, starting from age 11 years in comparison with younger animals, and eventually muscle cells start to be similar to heart cells. Heart cells have a lot of mitochondria. These mitochondria, they are dark brown, and this is why if you buy heart or liver, these cells are full of mitochondria. They are hyperactive. Compared to muscle meat is red, but heart is very dark brown. Okay, this shows that it has a lot of mitochondria. And in neck mole rats, The necmorets, they have many mitochondria, and with large volume of mitochondria in the muscle cells. Again, you have to understand why, because they change from using of glucose as source of energy, or at least part of the glucose as source of energy, exactly only to getting energy from lipid oxidation. Okay, this is how what is happening with muscles. This mitophagy drops, and the mitochondrial dysfunction is increasing, and imbalance in mitochondrial dynamics. Okay, and muscle mass and strength is dropping. So it's all why people are getting sarcopenia. But it can be stopped. It can be stopped if you do properly, everything properly. Muscles grow even in old age. I see it on myself. I mostly give exercises to my leg. I'm cycling and swimming in fins, so the muscles on hand, on arms are not very strong, not hyper size, not very big size. But okay, I should work maybe more on my upper body. But it works. I mean, muscle mass is increasing if you do everything properly. We can mimic in part the diet of young necmorets and blind morets by including large amounts of raw starch in our diet. Raw potato starch and raw tapioca starch are rich in resistant starch, more than 70%. Some kind of tapioca starch can have 80% by weight. But resistant starch is not digested in the small intestine, but some part is digested and converted to glucose. This is at least a danger. And you have to mimic old necmorets, not young. And if you will do this, like blind morets, they have a lot of fat, so you will accumulate fat if you consume too much of this raw starch. I discussed the inclusion of raw starch in our diet in a recent video, so you can check. And however, with increased consumption of raw starch, high concentration of glucose in the blood plasma may increase the risk of cancer development. This is what I was talking about. Also, part of this glucose is converted to adipose tissue. And as I have discovered, consuming more than 200 grams of raw starch daily may lead to accumulation of fat in subcutaneous adipose tissue. So what is the optimal way to increase the content of resistant starch in this raw starch? I don't know yet. Maybe it is no way. Maybe you have to limit consumption of raw starch to about 100 grams and replace it with, this is how necmorets eat tubers. And they don't eat the epidermis, the only inner part of the tubers. Instead of raw starch, we may include mushrooms in our diet as a source of insoluble dietary fiber. Also, edible mushrooms are rich in spermidine, a polyamine that delays aging in humans. Supplementation with spermidine reverses large artery stiffening, restores nitric oxide-mediated endothelium function, and reduces oxidative stress in arteries of old mice. So you see that, again, if you eat mushrooms, or even if you use supplementation of spermidine, it is very beneficial for your endothelium layer, for your lymph and blood vessels. In rats, spermidine reduced blood pressure and prevented cardiac hypertrophy, thus delaying the progression to heart failure. In humans, high levels of dietary spermidine are correlated with reduced blood pressure and lower incidence of cardiovascular disease. Okay, from my experience, eating about 1 kilo of oyster mushrooms combined with about a dozen high-quality eggs from free-range chicken is very beneficial for my health. This is what mushrooms are rich in spermidine. It is seasonal mushrooms, but to rely on seasonal mushrooms may be not a good idea, because you have to consume mushrooms every day, better. But okay, so if you start to include mushrooms or spermidine in some midwife, then your body function is improving. Okay, because otherwise lipofuscin, this is waste product, is accumulates, and oxidative stress is growing, fibrosis is growing, so you are aging. But if you spermidine can stop it, can reverse autophagy and can reverse your health almost to what you had in a younger age. In mushroom-based diet can support healthy gut microbiome. Inclusion of mushrooms in the diet of healthy adults led to significantly higher levels of intestinal short-chain fatty acids such as butyrate and propionate, and improved systemic immunity compared to the control group. Okay, so you can read the publications. Butyrate proportionally made up 53% of the total short-chain fatty acids upon feeding the high-dose mushroom blend to the healthy young individuals with average age 25.8 years. But amounts is ridiculous. High-dose mushrooms, it's only 15 grams of fresh mushroom. It is nothing, okay? If I was eating 1 kilo of mushrooms, this makes difference. But even 15 grams can work. So you see that again, if you eat at least a little bit of mushrooms, the production of butyrate and short-chain fatty acids is increasing compared to control group who are not eating mushrooms. Oyster mushrooms are rich in chitin and glucan prebiotics, which enhance beneficial gut bacteria activity, inhibits antibacterial, antiviral, antifungal effect, and provide potential benefits for athletic sports performance and recovery after intense exercise and cardiovascular system function. Yes, this is very beneficial. Oyster mushrooms, very easy to grow. I was growing more than 1 kilo a day, more than 1 kilo. It was easy. It was easy. It is not just, again, if you stay in countryside, growing, keeping hens or any birds like ducks and geese to produce eggs, it is easy. It is easy, guys. And also, I was growing mushrooms, these oyster mushrooms like this, or like this. It was very easy, pardon? It was very easy, and they were very delicious. If I try to buy at market, not the same quality. So I made it, the technology to grow mushrooms much better. I lost this opportunity now, but I probably think about how to recover, maybe not 1 kilo a day, but at least some amount to grow myself instead of to buy at market. At market it is not cheap, it is first limitation, and second, it is not the same quality what I used to produce. In my videos and some presentations, I attributed the longevity difference between microbats and megabats to the consumption of chitin in fine insects, and chitin can be fermented more effectively than fruit fibers in the large intestine. Unfortunately, I overestimated this impact. This what at least mind that I was trying to explain the difference. Also, microbats rely on lipids, while macrobats use sugars in fruits as the main source of energy. So similar to humans, microbats cannot digest all protein in insects. However, due to the high aerobic activity during flights, colonic transit time in bats is short, can be about 12 minutes only. So you see, and undigested protein is excreted with feces. So this is a way, you see, you should avoid these pathogens, these pathogens in your colon. So in short transit time is very critical. Maybe not 12 minutes because humans cannot do it so quick, but at least shorten as much as possible. In case with large colonies of microbats, air is saturated by ammonia produced by bacteria in their feces. In regular aerobic exercise, intake of resistant starch and mushrooms and mild laxatives such as megadoses of ascorbic acid, Epsom salt, and lactulose can help shorten colonic transit time. Okay, you see this is a gastrointestinal tract in different bats. This is bats eating insects. These are bats eating fruits. These are bats eating blood. This is so-called blood-feeding bats, vampires. So as you see, the stomach is relatively large in insect-eating bats because they rely on consumption of protein and also on consumption of the fat, but gut is relatively short. In fruit-eating bats, they have very small stomach. The gut is also relatively short, and so you see, but again, you see, they have no place for fermentation of the chitin. Unfortunately, so I overestimated, unfortunately. So chitin can be, in our gut, it can be fermented, and it will have a positive impact. But in bats, it is, they just don't have this colon, no have very small, this anus actually. They have no colon, no fermentation. They just go quick. Okay, series of biological agents fall into two broad cases: programmed longevity, this is gene theory, Hayflick limit theory, epigenetic theory, autoimmune theory, endocrine theory, and damage theories like wear and tear theory, membrane theory, mitochondrial decline theory, rate of living theory, cross-linking theory, free radical theory, gene manipulation theory, gene mutation theory, inflammatory theory, and the waste accumulation theory. You can check. And I recommend you to read the source of information. Quite good publications, I would say. Yeah, but many theories of aging are wrong, and popular anti-aging interventions are useless, especially expensive ones, absolutely useless, and moreover, they're dangerous. In particular, there are many pieces of evidence that transfusion of young blood and or blood plasma or exosomes, these extracellular vehicles, as well as utilizing stem cell therapy, may not only fail to achieve a healthier, prolonged life, but also elevate the risk of cancer development and potentially reduce short lifespan. This is what is happening. They get some, for a week or maybe maximum 2, some improvement of, maybe it is just expectations, it is not really based on chemistry and impact of this stem cell therapy, but this effect, again, if you check, I know you can check some people who, in the internet, people who promote this stem cell therapy, and you will see that after a while, these people look older than before the treatment. So mind that it is not so useful. And again, you pay money for what? Okay, and these methods are extremely rude, extremely invasive, and why to pay so? It's huge money. Stem cell therapy, the cheapest is $10,000 American. The cheapest. It is not in America, it is like you go to Latin America to get the treatment, and then you pay so big money, and it is all in vain. Or injections of these extracellular vehicles. Oh, okay, guys, you decide. It is your money, you decide what to do. There is substantial evidence challenging the correctness of the gene theory and epigenetic theory of aging. For instance, partial genetic reprogramming with Yamanaka factors demonstrated only 12% increase in the median, not maximum, median lifespan of wild-type mouse. Very low, very little, and actually it can be error of the experiment, just sometimes it is error, and it is very little increase in the lifespan. And meanwhile, doxycycline used in the genetic reprogramming can increase lifespan. This was not because of Yamanaka factor. Oh, Yamanaka factor, this Yamanaka is so smart person, he is a Nobel Prize winner. Guys, this is not fucking science. Sorry for rude words, but I'm a scientist myself. At least I have experience and can separate some people who are true scientists, like Nikola Tesla, or Linus Pauling, from people who just nothing. This is just advertisement. I will talk about a little bit about this. I was talking about this already. This all the industry of anti-aging industry is so corrupted, so disturbed. It is not science. It is the same like medical professionals are corrupted people, mostly corrupted people, and this is a business. This has no connection to science at all, zero. But anti-aging industry even more. So, okay, what else? The use of doxycycline or minocycline considerably increased median and lifespan in premature aging mice. This is well known. So doxycycline in low doses, like 20 milligram a day every day, or maybe 40 milligram a day, taken twice a day, is really very helpful. Some people use it for years, and it is suppressing chronic inflammation. It works absolutely amazingly well. Okay, precursors of nicotinamide are adenine dinucleotide, which is important for energy production. This is precursors like nicotinamide riboside and nicotinamide mononucleotide. This is too expensive supplements. Have been proposed as potential anti-aging drugs. Again, has no connection to reality, no, zero. By the way, I was checking nicotinamide mononucleotide a long time ago. I got it from China and discovered that first it is chemically unstable. If you don't keep it in a freezer, and even if it is, they are sending it to you by post, and it is already during this exposure to room temperature, it is already degrading to nicotinamide, which is cheap. And why to use expensive nicotinamide mononucleotide? I don't understand. Okay, research revealed that nicotinamide riboside administration resulted in 3% reduction in the lifespan of mice. How it can be anti-aging medicine? No way. Moreover, nicotinamide riboside and nicotinamide mononucleotide quickly degrade in the blood to nicotinamide. Again, you consume it, and already in your stomach it starts to degrade, and then in the blood it is also all completely decomposed to nicotinamide. Why to pay money instead? Nicotinamide is very cheap, but also it is going to bile because it is too much, and liver remove this nicotinamide from blood. It goes to bile, and in the bile it is deamination, so removing of ammonia, and by removing to ammonia it is getting converted to nicotinic acid. Nicotinic acid is helpful, but not as precursor of the NAD+. No, nicotinic acid is good for you to push you into deeper ketosis and to remove toxins. This is to remove toxins, not to increase energy production. You cannot increase energy production in conditions of chronic hypoxia and no aerobic exercise and compromised blood flow. This guy promotes, this guy is from Harvard. Harvard, I think, it is so stinking. It is, again, somehow they got like top university in the world. They paid big money for this, I believe, to get this #1 position. And this guy is David Sinclair. I have video about him. He is a trickster. He is not a scientist. He is a liar. He is multi-millionaire. Yes, he is very good in this convincing people to give him money. But look who is he. He is not a scientist at all. what he is doing. Okay, here at this picture, he is 47 years old. Yeah, and 47, he looks like 57, I would say. But what he made then, he's in the age 51. He used Botox injections like these women do in the age of 50. And he coloring his hair. It is shameful. I believe for a man it is shameful. He used also some hair implants. Okay, at this picture you again can compare. He tried different colorants, different dyes. So it's again, here it is again 51. He used such color, and he has gray hair. You see, he gray here, here and here, everywhere. Compared, it's funny, yeah. And here, his choice of the color was absolutely ridiculous. Under ultraviolet light, you see fluorescence of his hair, which indications that he used this coloring of his hair. It is shameful. What kind of scientist he is? He is claiming that his heart is as good as the heart of the 20 years person. Give me the data, show me, blah, 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 everybody can say. But he looks not younger. He is 47 here, and he is older than these cosmetic procedures. Okay, women do it the same. Some women make Botox injections every 2 weeks or 2 months. Okay, guys, so look, you can go to the social information. I finish this first part of the presentation, then I will continue in the next video, the second part. But again, mind that all these very expensive procedures, they are harmful. Don't do it. Don't think that these people like these guys are like gods. And he is promoting this Yamanaka factor. Oh, Yamanaka factor is, we will make genetic modification of humans. Guys, you already will have experience of genetic modification of humans with this new mRNA vaccine. Results are terrible, absolutely terrible. People die in age of 20, 25, 30 in China, especially in China, and it is only beginning. The further it will be more. Okay, so you see that. Okay, you can go to. I recommend you to read this. At least some of these publications to understand, if you want to understand. Again, if you have time to understand, if you have education to understand it, okay. If you don't, okay, follow your medical physician, your official dietitian. I don't care. I do this research mostly for myself. I see some improvement, but unfortunately, in biology, everything works slowly. I see that good results, and I will talk about this in next part, but it's hard. Okay. Okay. Thank you for your time. Again, don't forget to like this video and subscribe to my channel. And I would say I'm already at the stage that I'm finishing the theoretical part. At least I have to demonstrate the rejuvenation, so it will take time. I don't have time any longer to produce so detailed and review of literature, but also everything is clear now for me. So I need practical results, and I have to stay focused on practical stuff like to grow mushrooms again, because I know how to do it. I have to organize this growth again, consume more mushrooms, and also I have big house, 100 square meters here in Thailand, and I have sauna, but it is now used as storage room. I have to clean it. I have to prepare it and have to make sauna again, and I have to make sauna irregularly. It is a little bit not easy, so but it's life. It's life. Actually, it's good, very good for health. So take care. Thank you for your time again. Ciao, ciao.